The year is 1913. The word character carries a weight we can barely fathom today. It speaks of morality, of social standing, of inherent worth. It's a concept deeply intertwined with a burgeoning scientific movement, eugenics. This movement, now widely condemned, believed in improving the human race through selective breeding. Eugenicists, often from privileged backgrounds, saw desirable traits like intelligence and morality as purely hereditary. They argued for policies that would encourage reproduction among those deemed fit and discourage or even sterilize those considered unfit. This dangerous ideology took root in prejudices against the poor, disabled, and marginalized communities. The very idea of character became a tool for discrimination. It fueled forced sterilizations and discriminatory immigration policies. The shadow of eugenics reminds us of the ethical tightrope walked by any scientific endeavor that claims to define human worth. The legacy of eugenics serves as a stark warning. It reminds us to approach scientific advancements, particularly those related to human genetics, with caution and a strong ethical compass. The human story is not written solely in our genes. It is shaped by a complex interplay of factors, a truth that eluded the simplistic and dangerous worldview of the eugenics movement. The tug-of-war between nature and nurture is as old as the study of human behavior itself. Are we products of our genes, our destinies predetermined by the DNA we inherit? Or are we shaped by our environment, molded by our upbringing and experiences? Eugenicists, fixated on heredity, represented one extreme. They believed that social ills like poverty and crime were rooted in flawed genes. On the other side stood sociologists. They argued that social factors, poverty, lack of education, and discrimination were the real culprits. This debate raged on, often ignoring the possibility of a more nuanced reality. It's not a case of either or, but rather an intricate dance between the two. Our genes provide the building blocks, the raw materials of our being. But it's our environment that shapes these materials, influencing how our genes are expressed and how we develop as individuals. Understanding this interplay is crucial. It reminds us that we are not simply slaves to our genes. We have the capacity for change, for growth, and for shaping our destinies through our choices and interactions with the world around us. Imagine trying to decipher a language written in letters too small to see. This was the challenge faced by early geneticists. They knew that heredity existed, but the mechanisms remained shrouded in mystery. The invention of the electron microscope in the 1930s changed everything. This groundbreaking technology used a beam of electrons instead of light to illuminate specimens. It allowed scientists to visualize objects previously invisible, magnifying them thousands of times. For the first time, the intricate structures within cells, including chromosomes, came into sharp focus. The electron microscope unveiled the hidden world of genetics. It provided concrete evidence for the existence of chromosomes, the thread-like structures that carry our genes. It paved the way for understanding how genetic information is stored, copied, and passed down through generations. The ability to see the building blocks of life marked a turning point. It propelled the field of genetics forward, laying the foundation for the remarkable discoveries that would follow. It brought us closer to understanding the language of life itself. Our journey through the history of genetics begins with a humble monk named Gregor Mendel. In the mid-1800s, Mendel conducted meticulous experiments with pea plants. He observed how traits like flower color and seed shape were passed down through generations. Mendel's work, though unrecognized in his lifetime, laid the foundation for modern genetics. He identified the basic principles of inheritance, demonstrating that traits are passed down in discrete units, which we now call genes. He showed that these units come in pairs, with offspring inheriting one from each parent. Fast forward to the 20th century, and the discovery of DNA as the carrier of genetic information revolutionized biology. Scientists deciphered the structure of DNA, the elegant double helix that holds the blueprint of life. This breakthrough opened up unprecedented possibilities for understanding how genes function and how they contribute to our traits. Today, the Human Genome Project stands as a testament to how far we've come. This ambitious endeavor successfully mapped out the entire human genome, providing a comprehensive catalogue of our genes. This genetic map opened up new avenues for understanding human health, disease, and evolution.